So I'm very happy to introduce Sahiti Gowardhanam, who's going to talk about bacteriophage and how totally cool they are, and tell you a little bit about what we do in our course and why it matters. Okay.
I'm going to start by sharing my screen real quick. All right. I hope everyone sees my screen. Perfect. Hello, everyone. My name is Sahiti Gowardhanam, and I'm here on behalf of the MCDB 1161 Phage Genomics Lab. And today I will be talking to you about what we've been doing in our lab the entire semester, which is isolating and characterizing novel bacteriophage. I will be focusing more on the characterization of bacteriophage and also talk to you about the importance of bacteriophage in today's world. So to start off, what are bacteriophage? Bacteriophage are these guys in green, and they are a very specific kind of virus that infect bacteria. And bacteriophage, or most commonly known as phages, are one of the most abundant entities on planet Earth and can therefore be characterized based on many different criteria. Today, I will be talking about three criteria, which are life cycles, structure, and the genomic similarity. So to start off, life cycles, phages can go through two different life cycles, a lytic life cycle and a lysogenic life cycle. So in the lytic life cycle, you have the phage attaching to the bacterial cell, injecting its DNA into the bacterial cell, and then replicating and producing phage proteins, which are then assembled into mature phage particles. And these mature phage particles break down the cell wall and lyse out of the cell wall. So that is the lytic life cycle. Bacteriophage can also go through the lysogenic life cycle. So in the lysogenic life cycle, you have the bacteriophage attaching to the bacterial cell, injecting its DNA. So here, instead of replicating its DNA, what the bacteriophage does is integrates its DNA into the bacterial cell chromosome. And so as the bacterial cell replicates and divides, the phage genome is going to replicate and divide along with the bacterial cell. And due to certain circumstances, such as environmental stress, such as like UV light, the bacteria can break away from the bacterial chromosome. And after it breaks away from the bacterial chromosome, it's going to go back to the end the same way as the lytic life cycle by replicating the DNA, producing phage proteins, and assembling into mature phage particles and lysing out of the bacterial cell. So there are two different types of bacteriophages, a lytic phage and a temperate phage. A lytic phage can only go through the lytic life cycle, and a temperate phage can go through the lytic life cycle or the lysogenic life cycle. And a temperate phage usually makes this decision based on the environmental condition of the bacterial cell, inside the bacterial cell and outside the bacterial cell. So here on the screen, what we have are bacterial lawns with the phage infection. So the first one here is Barbie Things. It's a lytic phage, and you can tell that by the clear edges, and this is because lytic phages kill the bacteria. So therefore, the clear edges seen on this plate right here. So here's Candidum. Candidum is a temperate phage, and in what temperate phages essentially do are they can either, like I said earlier, they can either choose between the lytic life cycle or the lysogenic life cycle. So the cloudy areas are the phages that chose the lysogenic life cycle because they integrated their DNA into the bacterial cell. And so they don't kill the bacteria, and instead they give immunity to the bacteria from other phages. So you can tell that by the cloudy edge or the turbid edge. So the most common type of bacteriophage are tailed phages, and they have a head, which is an isocohedral, meaning 20-sided protein capsid, which contains the genetic material. And they have a tail that consists of the tail fibers as well as the base plate. The tail fibers and the base plate help the bacteriophage find its specific host cell and attach to the host cell. These are shown here are the three most common types of tailed phages: a myoverde, a cyphoverde, and a podoverde. A myoverde have short and contractile tails. Cyphoverde have long, non-contractile tails, which are very flexible, and podoverdes have short, stubby tails. So you can tell the structure of your bacteriophage looking at the electron microscopy picture. So here are Dorcas and Candidum, which are two bacteriophage isolated in our lab this semester. Dorcas is a myoverde phage and Candidum is a cyphoverde phage. Here's Burabura is a podoverde phage of the Microbacterium genus. We are yet to find a podoverde of the Mycobacterium genus. Phages can also be classified into clusters, which is based on their genomic similarity. Two phages in the same cluster have around 50% genomic similarity, and clusters can further be divided into subclusters, and two phages in the same subcluster have around 70% genomic similarity. And you can make predictions about what cluster your phage belongs to with an experiment called restriction digest. So in this experiment, you have the restriction enzymes mixed with the phage DNA. So how these restriction enzymes work are they bind to the DNA, and each restriction enzyme recognizes a specific DNA sequence. And as they go along the phage DNA, when they recognize that specific DNA sequence on the phage DNA, they make a cut and they move on. So at the end of the reaction and after you run the gel, this is what you would expect to see on the gel. And you can then take this and put it into a tool called Phage Enzymes tool. So here what you're doing is counting the number of bands that you got for each restriction enzyme. And once you put it into this tool, you get cluster predictions as seen right here. And what this concept is that you would expect phages with the same DNA sequence to have similar number of cuts. And that is just what this tool is projecting. And so now I would like to talk to you about the importance of bacteriophage. Why do we even care about them? Why do we even need to research about them? And that is because of the increasing antibiotic resistance in today's world, which is one of the biggest global threats that we have. And antibiotics are a way to prevent and treat bacterial infections. And because of the overuse and misuse of antibiotics, what it is doing is creating an environment where antibiotic-resistant bacteria can essentially thrive and grow and increase. So right here is an example. We have the E. coli bacteria over the span of 10 years against 5 different antibiotics. And as you can see, there's a clear increase in the resistance by 2008 over the span of 10 years. And because of the increase in antibiotic resistance, what this does is that we cannot treat bacterial infections as effectively as we used to treat, or not treat them at all. This is where bacteriophage come in. Bacteriophage can essentially replace antibiotics in a process called bacteriophage therapy. So here is a 56-year-old male patient diagnosed with disseminated cutaneous Mycobacterium coloniae. Disseminated cutaneous essentially means that he has abnormal looking tissue on his skin, and Mycobacterium coloniae is a very rare and chronic infection that is seen in immunocompromised patients. And so this is the infection for this patient is localized on his left arm. And what the medical team did is they administered bacteriophage therapy to this patient. The bacteriophage that they used in the therapy is Muddy. Muddy is one of the bacteriophage that is isolated as part of this C-phages program. So here is the before, how the infection looked on his left arm, and here is the after the bacteriophage therapy. You can see that there's a lot of redness that has gone down, and here is a scan. So here on the left arm, you can see the black dots, which is the infection, and after the phage therapy, you can see a clear decrease in the black dots, meaning clear decrease in the infection itself. So bacteriophage therapy right now is of compassionate use, meaning that it is only used when a patient's life is in danger and there's no other treatment for the patient to be administered to the patient. And this is why we need to do more research on bacteriophage, because bacteriophage has a lot of potential. And so with more research and increasing our knowledge on bacteriophage, we can use bacteriophage more commonly on a daily basis instead of antibiotics, which can help the global threat that we have of the increased antibiotic resistant bacteria. And yep, that is the reason why bacteriophages are so cool and so important. That is it for my presentation. Thank you so much for listening. I am now open to any questions that any of you may have. Okay.
Okay, you have some questions coming in. We can moderate that for you.
Yeah, that would be great.
Sydney has a question. Sydney, you're now unmuted, so you can go ahead and ask.
Hi, do you think that with further research and getting more bacteriophage in the system, that will this type of medicine become more accessible? As in, right now you said we can only use it if someone's like at death's door. Do you think we might be able to get it to where it could be prescribed instead of antibiotics?
That is what we want to do with bacteriophage, but right now we have very limited knowledge. So bacteriophage are very tricky, and when we use it on a patient, we need to tailor it so that it's made for the patient, and that's a very lengthy process. So just knowing more about the bacteriophage and how they behave and increasing our database with many bacteria, many different types of bacteriophages, I do think that we can get to a point where we can use them instead of antibiotics. But of course, we're very far from it. We still need to do a lot of research.
All right, cool, thank you.
Okay, we have a question in the comments. Emmett asks, does the morphology of a phage relate to any of its other features?
The morphology of the phage is very important when you're looking at the phage, using it in terms of phage therapy. Bacteria, we only use phages that are lytic for bacteriophage therapy, because what temperate phages can do is they can horizontal gene transfer, that is, and like I said earlier, they can essentially make the bacterial cell resistant to other phages. So we don't want that to happen. We don't want the bacterial cells to be antibiotic resistant because of the phage. So when we're looking at the morphology, we are looking at lytic phages. So that those are one of the most, those are what we're focused on when it comes to bacteriophage therapy. That answers the question. I see questions. Do you want me to?
I'll unmute Sarah Francis because she's been waiting with her hand raised. So Sarah Francis, you can go ahead. You're unmuted now.
Hi. Okay, so my question was, you were talking about kind of the phage therapy and stuff like that. So in your class specifically, how is the research you're doing kind of like pushing towards farther development of phage therapy? Like, how is understanding the clusters and the type, like, is it just for the database, or does it like help aid in farther development?
So it does help with the overall view of the phage therapy. We're definitely isolating bacteriophage and adding it onto the database, which, so if in the case of the 56-year-old male that I was just talking about, that was diagnosed with disseminated cutaneous, he was administered with muddy, which is a bacteriophage that was isolated by this program. So it was in the database. And so when they came across this case of this man, so what they essentially did is go back to the database, look at the different phages that we have that are in the database, pick back phages that are similar to the bacterial strain Mycobacterium coloniae, which is Mycobacterium schmegmatis, which is what we use in our lab. So they used this database to essentially find the match. So of course, in our lab, we're finding new bacteriophage and adding to the database, finding the morphology, but it helps the just general phage therapy because we're adding more to the database, which can then be used in phage therapy. They have more options to do the phage therapy. I hope that answers your question.
Perfect. Thank you.
Okay, and we have time for one more. I'm going to summarize. A couple of people asked this in the question and answer section. They're asking about antibiotic resistance. Is there a chance that phage therapy would lead to bacteria that are resistant to that entire form of therapy through whatever mechanisms?
I'm sorry, I didn't get. Is there a chance that phage therapy can?
Is there a chance that patients who receive phage therapy, is there a chance that the bacteria that infect them develop the resistance to the phage therapy itself?
Okay. I am not really sure. I haven't really come across anything like that. But one thing I can say is that bacteriophage therapy is a very lengthy process. So in the case that I was talking about in my presentation, that patient had to go through months of bacteriophage therapy, and it is definitely constantly administered. And of course, that could be one of the concerns that we have if they do, get resistant to the phage itself. But I am actually not really sure. I haven't come across anything like that in my research.
Yeah, it's possible it hasn't been used long enough to start to see it.
Exactly.
But it would be interesting to see if the bacteria themselves develop resistance to phage therapy. But it certainly seems like at the moment, we're probably not seeing it. It's good.
Let me chime in, Pam. The phage bacteria can develop a resistance to the phage, but the phage can co-evolve and then develop a different strategy to infect that bacteria. So there's sort of a play back and forth. You can't come into my cell and the phage says, oh yeah, I'm going to find another way, that kind of thing.